Hi, everyone. I'm Jeremy Wilkinson. I'm PacBio's segment lead for Microbial Genomics, and I'll be the host and moderator of today's webinar. I want to welcome you all to this webinar with today's topic focused on the recent advances by PacBio in microbial genomics with the release of new products, the HiFi prep kits for use on PacBio long-read sequencing platforms, as well as an example study comparing same sample microbial whole genome sequencing from PacBio HiFi sequencing utilizing the new HiFi Plex Prep Kit 96 compared to short read whole genome sequencing for assembly and antimicrobial resistance results analyzed on the Palmona Pathogenomics platform. So we have a lot of great material to cover today, and the presentations portion of this webinar is pre-recorded, but it will be followed by a live question and answer session with all the speakers. You are welcome to submit your questions at any point during this webinar by typing them in the area provided on your attendee control panel, and we'll get to those either in the chat or at the end during that session. We've also uploaded a few complementary pieces of literature to your control panel, so please feel free to download those and take them with you. We'll be recording this webinar and making it available for you in the next few days, so please keep an eye out for a follow-up email with a link to that recording. Immediately following this webinar, you'll receive a brief questionnaire. Please do take the time to fill that out, as it does help us understand your needs and plan for future webinars. You can also stay up to date with on upcoming webinars by visiting pacb.com/events. So we have some important audio information. So this webinar, like I said, has both live and pre-recorded content. If you do encounter audio issues as we switch between the live conversation, which we're happening right now, and the pre-recorded talks that are about to start in this webinar, you may have the wrong audio mode selected. So please check the audio tab to ensure that you have selected the correct one, either computer or phone, depending on the device you're using. If you do continue to experience audio issues, we do apologize for that, but we are recording this webinar and we'll be making it available in the next few days. If you are attending on a phone, the audio quality is better if you're using a headset or headphones. We also currently have active promotions for our long-read Revio system. So if you're interested in more information on these promotions, please let us know in the post-webinar survey or by going to the Contact Us form on pacb.com. So for the agenda today, this is a 1-hour webinar. We have 2 talks and a live Q&A at the end. I'm joined today by 2 great experts. So the first is Gregory Young. He's a senior product manager of whole genome sequencing at PacBio. He's going to be presenting on more samples, lower cost, less time, using new PacBio HiFi prep kits, plus microbial whole genome sequencing and antimicrobial resistance. Second, we have Jonathan Monk. He's the co-founder and head of bioinformatics and analytics at Palmona Pathogenomics. And he's going to be presenting on comparing short read to PacBio long read sequencing results in the Palmona Pathogenomics platform with applications in microbial whole genome sequencing, antimicrobial resistance, and phylogenomics. So with that, let's get started.
Thanks, Jeremy, for the introduction. And I'd like to thank everybody for attending today's webinar. I'm going to start off by talking about HiFi sequencing, talk a bit about the HiFi sequencing workflow, and then I'll jump into talking about our new HiFi prep kits. PacBio HiFi reads combine the best of 2 worlds, high accuracy with long read length. So you get the accuracy of a short read, but at 100 times longer length. Now this is possible because we make multiple sequencing observations on the same stretch of DNA from a single molecule. This washes out any random errors and allows for a highly accurate consensus sequence to be called from the subreads. That consensus sequence generation happens on the instrument, so it's no additional expense to the customer to run that on your local server or HPC system. If you want to explore our HiFi data and see for yourself, we have many data sets available. I encourage you, definitely if you're working with metagenomic or microbial applications, to visit our PB Metagenomics GitHub page. We also have other data sets available for both the Revio and Sequel II systems at pacb.com/datasets. The first step to creating a HiFi read, or for any long read DNA sequencing experiment for that matter, should be the extraction or isolation of high molecular weight DNA. The first step in preparing a HiFi library is to mechanically shear your DNA. For most whole genome sequencing applications, we recommend an average fragment size between 15 to 20 kb. When working with more degraded samples, smaller average fragment sizes may be necessary. The key here is to really have a narrow distribution. You don't want too many short fragments, and you don't want too many really long fragments in order to optimize that coverage and sequencing yield. Once you have your DNA fragmented, the sample will be brought through a DNA damage and end repair step, then ligated with our SmartBell adapters, and this can include using an indexed adapter. And then that will be treated with a nucleus cocktail to remove any unligated, semi-ligated, or damaged DNA sequences. After the nuclease treatment, the library is cleaned up using beads or using an optional size selection method to further enrich for larger fragments. Now, prior to loading your library on the HiFi system, the library needs to be bound with a sequencing polymerase. And then finally, once you have that polymerase bound library, it's loaded onto the HiFi sequencing system. Previously, bottlenecks in the workflow and the prep costs were barriers to scaling long read sequencing or multiplexing many samples onto the single sequencing chip. Now, these bottlenecks were primarily around available options for DNA sharing and size selection. In terms of cost, the need to purchase single-use individual consumables for DNA sharing and the cost on the library prep reagents made large multiplexing experiments prohibitively expensive. I intentionally put the emphasis on were because we now have solutions that eliminate these bottlenecks and dramatically reduce the library prep costs. And these solutions are in the form of the new HiFi Prep Kit 96 and the new HiFi Plex Prep Kit 96. Now, the HiFi Prep Kit 96 is designed for high throughput workflows for doing human, plant, or animal whole genome sequencing projects. It includes a scalable DNA sharing solution. The workflow itself is going to take 60% less time than our previous recommendations, and it's going to have a 40% lower overall workflow cost. Now, every step of this workflow can be automated for that end-to-end solution. But today, in today's talk, I'm going to focus on the HiFi Plex Prep Kit 96. With the HiFi Plex Prep Kit, we have a new scalable long-read multiplexing workflow that's going to be very cost-effective for our customers. The library prep reagents are priced at $32 per sample, but require separate purchase of one of our 4 SmartBell adapter index plates at $7 per index, for a total library prep cost of $39 per sample. Now, if you're looking to really scale up, it is possible to do 1,500 samples in a single Revio run. And this is by doing 4 cells at a time, each cell with a 384 plex. This kit and workflow represents a more than 50% reduction in our per sample cost over our previous solution. And one can prepare up to 96 samples in as little time as 6 hours. Another really cool feature is that you can go in with less than 300 nanograms of DNA per sample. Our previous solution, this was the minimum amount of DNA we recommended. Here is the maximum amount of DNA that we recommend going into. Now, this kit and workflow is designed for microbial whole genome sequencing or for metagenomic shotgun sequencing where you may want to do functional or taxonomic profiling, or for amplicon sequencing where you need an adapter index solution. Now, this workflow can be automated, and we do have methods available on the Hamilton NGS star method today. One of the ways we've enabled these workflows to be scalable is the development of new low-cost, high-throughput DNA sharing methods using liquid handling automation. We have qualified protocols for the Microlab Prep and the NGS star systems. The Microlab Prep is really cool because it's a low-cost, small benchtop system. It can shear 24 samples in 22 minutes, while the NGS star is a higher throughput system. It can do 96 samples in 10 minutes using the multi-probe head. PacBio can provide you with the method for the Microlab Prep directly, while we recommend going to Hamilton for proper installation of the sharing method for the NGS star system. What's also really great about this new method is that it doesn't require any additional individual consumables. So the cost is approximately only 12 cents per sample, and this is the cost of the pipette tip and plastic plate holding the samples. The method also produces really nice size distributions perfect for HiFi sequencing. As I've shown here on the right-hand side of the slide, the size distribution of fragments are nearly identical to those produced by the Megaruptor 3 system, which has been the standard over the last few years. Okay, for now, let's go into a little more detail on the HiFi plex workflow. So the workflow begins with that pipette DNA sharing method that I just discussed. After sharing, the samples are brought into library prep using the new HiFi Plex Prep Kit 96. For the first 4 steps, the samples are going to be processed in parallel and automated on the NGS star system. This includes sample pooling. But once you have the samples pooled, the cleanup and nuclease steps are performed manually. And this takes a total of approximately 6 hours. To then prepare the samples for sequencing, the 4 pools are combined for the primer annealing, polymerase binding, and final cleanup steps, which we abbreviate as the ABC workflow. We recommend 50 to 300 nanograms of genomic DNA input per sample. If you're working with amplicons, that should be reduced to 20 to 200 nanograms per sample. And since this is native single molecule DNA sequencing, base modifications can be detected by saving the polymerase kinetic data. Now, this kit and workflow supports a minimum plex level of 24 samples, and up to a maximum of 384 samples can be sequenced onto a single Revio SMRT Cell. With this workflow, up to 96 sequence-ready libraries can be built in just one single workday. With the combination of this new HiFi Plex kit and the Revio system, PacBio can now deliver a HiFi microbial genome for under $50. This puts us at cost parity with short reads, but with huge advantages in data quality and analysis that Jonathan here in a moment will discuss. At the 96 plex level, sequencing costs $10 per sample and $3 per sample at the 384 plex level. A 96 plex will consistently produce over 100x coverage per sample, depending on how you balance the pool. And considering that only 15 to 30x is necessary for a good microbial assembly using PacBio HiFi reads, users can easily scale up to 384 samples per Revio SMRT Cell. And finally, to finish up, I wanted to present the primary sequencing results for the data set that Jonathan will be discussing. This set of 96 escapee pathogens was brought through the HiFi Plex protocol in the Hamilton NGS star system, including the DNA sharing step. I also want to note that we did not normalize the DNA input because we knew the coverage would be high enough per sample to not have to worry too much about having the perfectly balanced pool. And as you can see, this protocol can produce amazing results for microbial sequencing. We have 12 KB reads on average with a median read quality score of Q40. Over 94% of the bases have a predicted quality of Q30 or higher, and 99.8% of the reads could be confidently assigned a barcode. So I just want to take a moment to let that kind of sink in. You can now get 12 KB reads at a Q40 quality for under $50 per sample. More importantly, though, it's all about what you can do with this kind of data. So I'm going to turn it over to Jonathan so he can discuss what they've been able to glean from this data set using their analysis pipeline.
Yeah, thanks, Greg. I'm happy to present here on comparing short read to PacBio long read sequencing results in Palmona's Pathogenomics platform P3, with particular applications in microbial whole genome sequencing, antimicrobial resistance, and phylogenomics. But first, I want to give an overview of Palmona. Our mission here is to empower medical and public health communities to understand, outpace, and outsmart pathogens, including those we've never seen before. And our focus is on building innovative products and services for bioinformatics, analysis, and surveillance, including our cloud-based platform P3 for bioinformatics and advanced analytics with dynamic visualizations and sharing, which I'll demo later today. Here's a brief outline of my talk. I'll start with a sample and sequencing overview. I'll next move on to a comparison of the coverage between these samples, an evaluation of the assembly quality, the impact of different assembly results on phylogenetic trees and epidemiology. I'll go into a comparison of AMR gene detection and the impact of that gene detection on AMR prediction results, and I'll have a demo of Palmona's P3 platform. Here's a brief overview of our samples. We had 96 extended spectrum beta-lactamase ESBL single colony isolates from the Escapee family, which include E. coli, Staph aureus, Klebsiella pneumonia, A. baumannii, Enterobacter, and Enterococcus. These 7 species have been identified by the CDC and WHO as emerging antibiotic resistance threats. And we collected these strains over 5 years at a regional public health lab. The samples were sequenced using both short read and PacBio long read technologies from the same DNA extraction, and they were de novo assembled, annotated, and evaluated using the P3 platform. The input DNA for the PacBio long read sequencing looked like this. We first sequenced these with short read technologies, and then we passed on that same DNA for the PacBio long read sequencing. And you can see at that point we had some samples that had a very low sample volume, down to 5 microliters, and some samples with really low concentration of 6.8 nanograms per microliter. Overall, the input DNA ranged from 120 to 606 nanograms. And Greg showed you this read distribution length plot before, but here it is again, just to emphasize the really significantly long reads that we got from this run, the average being around 12,000 base pairs in length, and some that extended to 30,000 and even 50,000 base pairs long. Briefly, I wanted to go through the bioinformatics methods that we applied to these reads, both for the long reads and the short reads. For the read QC and QA, we applied FastQC and multi-QC. For read trimming and filtering, we applied FastP. Assembly was the only tool that was applied differently from the long reads to the short reads due to the difference in their read lengths. We applied Fly for the long read assemblies and Unicycler with spades to the short reads. We annotated these genomes with PROCA. We performed AMR gene identification using AMR Finder, virulence factor gene identification with Abertamer, multiple sequence alignment with Sibelia Z, and phylogenetic tree construction with IQ-Tree. And I want to emphasize that all of these steps can be performed automatically in our P3 cloud-based platform for incoming sequence reads. Here's a demonstration of the mean quality scores for those reads. On the left, you see our long reads. And I want to emphasize the difference in x-axis length. You can see the reads range from 0 to 50,000 base pairs in length on the x-axis on the left, compared to the much shorter reads on the right that range from 0 to 150. And you can see that the Phred scores were really quite high for both technologies, but for the long read technologies, you can see that they started out near 40 and only started to have meaningful drop off after 25,000 base pairs in length. And even in that case, rarely went below Phred scores of 30. Here's a comparison of the coverage between those genomes, assembled genomes. And you can see in blue, the short read coverage. The average for those was 262x coverage. And in red, you see the PacBio coverage. The average there was about 182x coverage. So all of these genomes had really high coverage. We got a lot of reads back for these sequencing files, and we had really high coverage that passed any kind of coverage restrictions we might have had. And I want to emphasize that all the data for this coverage comparison can be exported from our P3 platform for further analysis and comparison like is done here. On the assembly quality side of things, after assembling these genomes, we compared the number of contigs that were produced. And you can see that for the short read assemblies, the number of contigs, the average number of contigs was 167, whereas for our long reads, the average number of contigs was 3. And it can be difficult to see those long read covered or contig counts there. Because they're so few, but the average, again, was 3 plus or minus 1. And so that's indicative of having one really high quality contig representative of the chromosome, and then maybe a few plasmids included in those assemblies. On the bottom, you can see N50 scores, and N50 scores are a proxy for assembly quality, with higher N50 scores being better. And you can see that the PacBio long read technologies produced N50 scores of around 5 million base pairs, which are basically covering the entire assembled genome, whereas the short read technologies produce N50 scores with an average of about 200,000 base pairs. And again, those can be hard to see in the bottom there, but they are there. So what does this mean for your final assemblies? We constructed bandage assembly graphs to visualize the impact of the different sequencing technologies. So on the left, you can see the result of an assembly graph for our long-read contig assembly, and you can see a really nice closed assembly for that chromosome there, along with a plasmid on the next to it. On the right, you see the contig assembly graph for short-read assembled genomes, and you can see that these are much more complicated with several different branching segments to the network and a couple other segments on the bottom there that were not possible to integrate into the final assembly. Here's another example. On the left, long-read assemblies, we have a chromosome with 2 plasmids. On the right, again, much more complexity. And finally, another example with nice closed contigs on the left and a complex assembly graph on the right. And what are the impact of these differences in assembly quality? One impact is for comparing genomes between strains in a species. And so we performed multiple sequence alignment using both the long-read and the short-read technologies. At the top here, you see the result from the long-reads. These long-reads really enabled detection of large-scale genomic rearrangements between bacteria, such as inversions, duplications, and translocations. The results for the short-reads allowed us to still compare these syntenic blocks. However, the relative position and the orientations of the genomes, of the contigs in the genomes, is lost. And this has impacts on things like phylogenomics, where we're comparing strains to each other for applications in epidemiology. And you can see here, we looked at bootstrapping, which is a method applied to a phylogenetic tree to determine the confidence in that calculated tree. And the tree calculated from long-read assemblies generally have branches with much higher confidence levels. As you can see on the figure on the right, the long-reads are in blue in this case, and the short-read bootstrap values are in red. And we can actually overlay those bootstrapping values to the tree itself. So as you see, the blue values are higher bootstrap, blue and black values are higher bootstrap values. And on the left, we have our long-read assembled tree. The regions of poor bootstrap values are much more localized to the ends of the tree, whereas on the right, we have large clades with poor bootstrap values. And so this can have impact on interpreting a phylogenetic tree and the similarities between strains in a species. So next, I want to start our demo of the P3 platform that allows you to perform a lot of the analyses that I just went through. P3 is a platform to deliver clarity to complex data in 3 easy steps. Users can upload their genomics files. They can run bioinformatics and AI-based analysis platforms in the cloud, and then they can experience dynamic visualizations and insights from that data. And the goal of P3 is to streamline the lab workflow and pipelines in real time, making identifying complex infectious diseases efficient and actionable. So I'm going to start the demo now. Okay, so here I'm logged into the Palmona P3 platform, and I'm welcomed with an analysis or a display of my recent workflows. And I'm going to start assuming that I had new data to load to the platform. I can go to the data module on the top here, and I can create samples. I can browse a sample sheet here, and I can upload a CSV or Excel file that has description of all of my samples in it. So in this case, I have samples on the left, I have the file name from the sequencer that comes back for those, and then I have associated information for those samples, including the source they were collected from, the collection latitude and longitude. I can also then drag and drop some of my sequencing files for those samples and have them populated in the system. And you can see that these red triangles change from red to a green check mark when the files have been uploaded to the system and staged for upload. Next, I'm going to show you how to take these samples and run a workflow on them. I can click on the Workflows tab and click on Select a New Workflow, and you can see here we have a PacBio assembly annotation for bacterial whole genome sequences from PacBio long-read sequencing. And I can click this button here to run that workflow and then visualize the results with the Visualization tab. I'm not going to run this workflow now because it's already been processed, but I'll demonstrate the results in the Visualization tab. I do want to emphasize that we have various different workflows in place here that can take different sequencing tools, including Illumina and Oxford Nanopore, but the demo I'll show today is for PacBio long-read sequencing results. If I click on the Visualization tab, I can see the output of a particular workflow, and here I'm presented with the number of strains that were processed, the number of gene families detected in those strains, as well as which of those were particular virulence factors and antibiotic resistance genes. If I'm curious about the quality of that run, I can click on the Multi-QC tab on the left, and I can scroll down to see some general statistics about the quality of the run. And so like I presented before, the N50 scores for these PacBio assembled genomes are quite high, around the 5 million base pair in length scores. I can also switch these long read assemblies to my short read results, and you can see that reloads the page. And in this case, I'm seeing short read results that are N50 scores with around 200,000 to 300,000 base pairs in length. And I can even scroll down and look at the results in terms of the number of contigs. And again, these are results I showed earlier in the presentation, but I just wanted to demo them in the platform. You can see for the short reads, the number of contigs is much higher than it was for our long reads. I can also use the platform to look at individual strains. So I can go to my table here and look for a particular strain of interest. I'll look at AEC24 right now. And this page will load with the specific AMR genes detected for that strain. Here on the left, you can see there are 18 different AMR genes, virulence factors, as well as some different phenotypes and other information about the strain. In this case, I'm looking at the short read assembled genome, and I know that because it's fragmented into several different contigs. You can see that it's broken up into all these contigs. I can zoom in and look at where some of these AMR genes were detected. You can see AMR genes are displayed in red, while virulence factors are displayed in that orange color. And I can see that some of these genomes have AMR genes. However, they're often on some of these small contigs, which can be hard to determine whether or not these genes are plasmidic or chromosomal. If I reload this page to display the long read results, this page will now update. You can see the number of AMR genes detected, virulence factors detected is the same, but my genome assembly is much higher quality, where I have a single chromosome here in the light gray, and I have the indication of a plasmid here in this dark gray. And if I zoom in, I can actually look and see that several of these AMR genes and virulence factors were actually detected on that plasmid. That was really difficult to do with my short read results. And I can zoom in, I can see some of these virulence factors. For example here, these are these IUC operon, which is involved in iron transport. Those are found on this plasmid here. And I can also see that the beta-lactamase itself that is causing the strain to be an ESBL strain is found on that plasmid here with that CTXM55 gene. Let's just take a look at one more example. I can, again, look at AEC14 in this case. This is a nice example because it had an example of a duplication. I can zoom in, again, it looks like a single chromosome and a nice single plasmidic contig here. I can zoom in and see a lot of these AMR genes on this particular plasmid. And I see my CTXM14 gene here on the plasmid. And if I scroll over, it was also detected on the chromosome. So this is an example of a duplication that might not be detected with short read sequencing technology. I'll go through a couple other features of the platform. Because all the data is input into a database, I can actually search it. And so I can look for strains of interest that, let's say, came from a particular source, like the bloodstream, and may have a particular AMR gene of interest, like, let's say, genes affiliated with cephalosporin resistance. I can click on the gene of interest, submit a query, and see a list of strains that had both that AMR gene and came from the bloodstream. I can also look at various different representations of all the different resistance genes found in my strains. So if I were to click here and look at another CMY, I can click on CMY2 here and see that that gene was found in 7 different strains. I can also look at the representation of these genes in heat map format, where I can go here, pick an AMR gene of interest or category of interest like cephalosporin resistance, and see these genes displayed in the heat map, where presence is indicated by orange and absence is indicated by black. And if I was interested in following up on one of these strains, I could actually click on it and follow it to the strain page there and look at the presence of that gene on that genome. I can also filter this heat map, again, by source. So let's say I wanted to know which of these strains were from the urinary tract, or, for example, the bloodstream. And these are dynamically filtered to show which strains have particular AMR genes and are from particular sources. Finally, I also want to demonstrate our phylogenetic tree construction. Again, for our long-read assembled genomes, this is the PacBio assembled tree. I can actually overlay information like the source where these strains were isolated from, and I can see the bootstrapping values. So here in this case, the blue values indicate higher bootstrapping values, indicating higher confidence in this tree, whereas the red values are some of the lower confidence values. And I can also see where these strains came from their various different isolation sources. I can also filter these here by, for example, name. Let's say I only want to see the strains that started with a 2, and those are dynamically filtered here. So that concludes my demo of the P3 platform. I hope I've demonstrated to you how P3 delivers clarity to complex data in 3 easy steps. The last thing I want to show is a comparison of these AMR gene identified between the two sequencing technologies. So on the left, I have a heat map of AMR genes that were detected in our long-read genome assemblies. And on the right, I have the same heat map, but these are for AMR genes detected in our short-read genome assemblies. As you can see on the left, the number of AMR genes found in the long-read assemblies was higher. There were 1,813 AMR genes found across these 96 strains compared to 1,766 AMR genes found in the short-read assemblies. And these were primarily in the case of duplications. So you see on the left, there are some dots there that are darker blue, indicating that there were multiple copies of some of these AMR genes, which I also showed you in the P3 platform. But you might be wondering, what's the impact of the difference in AMR gene calling between these two technologies? And here at Palmona, we actually build machine learning-based models to predict the phenotypic resistance of strains based on their genomes. And we did find two cases where these differences in AMR gene calls led to differences in those phenomic predictions. And one of the cases was for the strain AEC1, where the long-read assembled genome predicted strain to be resistant to ceftazidime, a third-generation beta-lactam, while the short-read genome was predicted to be susceptible due to the lack of a CTX-M15 gene in that genome. Similarly, in the AEC66 genome, our long-read assembled genome was predicted to be resistant to streptomycin, this aminoglycoside, but the short-read genome was actually predicted to be susceptible due to a missing aminoglycoside phosphotransferase. So those are two clear examples of where the predictive results were different based on the sequencing technologies and the genes detected using those two sequencing technologies. So in conclusion, I hope I've demonstrated to you that PacBio HiFi 96 Prep Kit produces high coverage genomes, even from low input samples without normalization. Those long-read PacBio genome assemblies are significantly more complete than short-read assemblies. The PacBio genomes assembled with long-read technology identify gene duplications and instances of genomic rearrangement that are messy or impossible to detect with short-read technology. Epidemiology applications are improved from PacBio long-read genome assemblies with more robust phylogeny results. The PacBio and short-read assembled genomes mostly align on AMR gene identification with some key distinctions that have downstream implications on phenotype prediction. And finally, that the P3 tool is a powerful way to analyze and visualize genomics data from both short-read and long-read assemblies. We'll be presenting these results at a couple upcoming conferences, including APHL in May, as well as ASM and ASM Micro in June. And with that, thank you all for attending, and please visit our website, palmona.com, to learn more.
Thanks. Thanks both for the great talks. Now we're going to move into the live Q&A session with both of the speakers. If you haven't already, please submit your questions in the area provided in your attendee control panel. All right, so we do have a few questions already that have been submitted, so thank you for that. First question that I will assign to Greg. What limits the sample number to 384 per cell?
Yeah, so some of that's just a function of the number of indexes that we currently provide. So we just last month, we launched 3 new SmartBell adapter index plates. Each of those plates have 96 adapter indexes. So now we have a total of 384 adapter indexes. Now with the throughput of the Revio system, as I highlighted in my portion of the talk, with a 384 plex, you generally get around somewhere probably between 15 to 30x coverage per microbial genome. And that's sort of where you want to be at to be able to get an assembly from HiFi data. If you were to increase that plex level on a per cell basis, you can still get a lot of useful information, but then your coverages are going to go, drop below that if you're trying to do assemblies. So we generally recommend keeping it at that 384. But with the power of the Revio, being able to do those 4 cells in parallel, you can actually probably do, if you want to increase, you just do multiple cells to run in parallel. Because at that point, the majority of your cost for that experiment is actually on the sample prep side and not the sequencing side. So, yeah.
Great, thank you. Another question for you, Greg. Is it okay to combine samples from different species on one cell?
Yeah, totally. And that's sort of what we've done here. I think the only thing to kind of keep in mind is like generally recommend if you're doing, microbial whole genome sequencing like we've talked about today, make sure you just pull, microbes together, right? You don't want to necessarily pull like a sample that may have a very large genome with a bunch of samples that have very small genomes, because then it's just going to just, really throw off that balance you're trying to get, because you want to make sure you're getting enough coverage on each of those. So you'll probably get a lot more coverage on the short stuff and maybe not as much coverage on the larger genome. So you just want to kind of keep that in mind when you're thinking about your pooling strategy.
Awesome. Yeah, also the data set that was presented today there are multiple species too. So another question for you, Greg. Is it possible to use the HiFi plex prep kit with Sequel 2? If so, what are the recommended DNA inputs for that? And for multiplexing, what is the expected performance on Sequel 2?
Yeah, I should have mentioned that in the talk, but so you could definitely use this kit for Sequel 2. So just in that ABC step, you would just bind with the sequencing polymerase for the Sequel 2 instrument. There, we recommend doing 96 plex as sort of the maximum, given that the Sequel 2 instrument doesn't quite have the higher throughput as the Revio system. And the expected results should be very similar to what you get on the Revio. The only difference would be just the lower throughput, right? You're just not going to get as high of a gigabase yield on a Sequel 2 system that you'll get on the Revio system. But the performance of the data should be pretty much identical. It's just that, your capability in Sequel 2 is probably pretty maxed out at 96 per SMRT cell, whereas the Revio enables you to scale that up a little higher.
Great, thank you. Another question for you as well. So what is a lot of great questions about the library preps, which is awesome. What is the expected or average recovery percentage after library prep using the Plex prep?
Yeah, so it's going to, vary a little bit depending on the quality of the samples put in, but it's generally between 10 to 20%, and that's on an individual and pool basis. So generally, that provides you, since we're pulling everything going in, plenty of material to run one or multiple SMRT cells. But yeah, it's somewhere around 20%, I would say, like on average.
Thank you. How many different samples can you process at once using these kits?
So we generally recommend doing 24 samples at once for each time, because that's sort of how we configured the kit to provide enough reagents to do like sets of 24. Or you could do like 48 or 96 or something in between. It doesn't have to be like a multiple of 24. We just don't recommend going below 24, or you won't have enough reagents to possibly fill out the whole 96 that you purchased the kit for. And some of this has to do around with expectations around automation, right? We've this workflow should be easy to automate. We have a qualified program on the Hamilton NGS Star system, but you could take those workflow steps. There's nothing preventing you from doing those workflow steps on another instrument if you had the resources to work on developing that on your particular platform, right? And so the dead volume we provide is enough to do 4 runs on an automated liquid handler. So that's why we say, like, you should do things at a minimum of 24 samples at a time.
Yep. Should look into here. We're getting a lot that are coming in now. What's the lowest possible nanograms loaded onto the Revio for 30 KB library? So it's probably 50 nanograms, right? That's the answer.
Well, we recommend 50 nanograms to guarantee it, but maybe I'll kick it over to Jonathan, because we did have like a sample on this, and in the data set he provided us that, we thought, he was like, oh, this is very low concentration. There's hardly anything in there. And, it may drop out. And we're like, okay, we'll just give it a shot anyway. And we prepped it. And I believe we got like a few thousand reads for it, and you were able to get a lot of data from it, right, Jonathan?
Absolutely, yeah, we have pretty nice coverage actually, even for that low input concentration, low input genomic content sample. Yeah.
Yeah, and I think when we measured the concentration, I mean, there was only like, it was definitely below 50 nanograms. There was like only
Oh,
probably a
way
few
more.
nanograms of actual
Yeah,
DNA there.
exactly.
Yeah.
Yeah.
Yeah. So it is possible. You just, yeah, you wouldn't, it also depends on like, in this situation, since we only had a 96 plex, right, we had sort of, we were sort of, oversampling. We had a lot of high coverage, right? So in that situation, you're more likely to detect that lower abundant species than say if we did a 384 plex, maybe we don't get quite as high coverage on that low input sample unless we kind of normalized everything down to that level, which we probably wouldn't really recommend doing. Yeah. But generally, I would say about 50 it should be where you sort of, if you have the DNA, it should cap it at 50, yeah, to guarantee that you get everything.
So the next one is the plot that you showed of the sheared DNA seemed to have two major peaks. What could be the reason for having those two peaks of sheared
I think they're probably referring to the HiFi read length distribution. It had like a little bit of a hump or shoulder to it. So that is just a product of the fact that we're doing 96 samples, right? So not all the samples are coming in with the same level of quality, right? So some of them are going to have a little more degraded DNA than others, right? So the others that have really good DNA, we're going to be able to shear and keep them at somewhere, that around 15 kb, size. But the ones that are already degraded already have a massive DNA that may already be below that. And that's just simply what we're seeing in the sequencing data, right? We didn't get everything perfectly within where like every single fragment is between 15 to 20 kb, right? It's more of a spread. And that's simply just what we're seeing in the data. So it's just a fact of just pulling 96 different samples together.
Yeah. All right. Thank you. Um, is it possible to use any of your kits for mycete WGS?
Yeah, so I'm sorry, I'm not too familiar with that. But is that a fungal species? But yeah, I mean, generally, anything that has a sort of a small genome size here, where you can multiplex 24 or higher on a Revio Smart Cell. I know, just for reference, I did have that one slide in the presentation that showed you sort of what the gigabase output of a Revio Smart Cell would be. So if we're talking like a 15 kb insert, that's like 90 gigs. So basically, you take that 90 gig mark, divide it by your 24, and if that gives you enough coverage on your genomes, yes, you can sequence it easily using this kit and using either the Revio platform, or you can do the same exercise with a Sequel 2 platform if you have access to that particular instrument.
Great. Thank you. So another question, we use fusion primers with custom index for amplicon library prep and use kit provided adapters to make a smart bell, but there's no adapter provided in the HiFi prep in the plex kit, which makes it a bit difficult. Do you have any comment on that?
Yeah, so we do, recommend, using, our index adapters, and that'll just simplify the process of doing the on-instrument demultiplexing because we've sort of validated that. We know there's not going to be any possible crosstalk with those barcodes or misidentification. So you can still do that with this kit, right? So I said, we're still talking about a $39 per sample cost, which is going to be your lowest option with any of the library prep kits we provide. So the fact that you do have your own index or barcode there on the sequences, it is still possible you can just basically dual index something, right? You can demultiplex it with ours and then do another run of demultiplexing with your barcode sequence. Or if you have, once you get the data and you have your own bioinformatic pipeline, you can just simply then use your index if you want. But having the PacBio index is not going to harm anything, and it's not going to, make you have to change anything about the sequencing experiment itself.
Thank you. Is it possible to do the pipette DNA shearing manually without using Hamilton instruments?
Yeah, unfortunately you can't. So the way this method works is it's a very fast up and down mixing step. at certain accelerations and decelerations and a certain volume. It's just not possible to kind of reach those speeds or doing this manually. So it is, great technique, but it is, currently, only done on the Hamilton instruments because they have that sort of parameter matrix of being able to pipette mix 300 microliters of volume, do it at the speeds that get this nice profile that we showed you, right? But the good news is like that microlab prep system is relatively inexpensive in terms of like laboratory equipment, right? So it is, not that much more than purchasing, say, like a Megaruptor 3 system or some other kind of DNA sharing technology. So I would really encourage people to look into, getting that technology. It can be, we can sell it, PacBio can sell it to you, or Hamilton can sell it to you. So I encourage people to check that out because it's not an expensive liquid handler, and it can do other things like our short read eliminator kit, which we recommend for doing larger genomes. You can automate that step on the microlab prep system as well.
Great, thank you. A question that I can answer here. Will the slides be available to us after this webinar? We will be sharing the recording. If you do want the slides, you can reach out and we can share those though, but we won't be sharing those in the standard process. But you can reach out and we're happy to share them. Back onto the loading question for the 30 kb library. Would it be 50 nanograms for the entire pool of 96, or is that per sample in a 96 plex?
It's per sample. Yeah, so per sample, you do that, and then it's per sample.
Yeah. And then the total loading would need to be what, at least a microgram or 2 micrograms?
To load on the instrument? No, usually generally, it all depends on the molarity, right? So we'd have to, since you have if you do have like a 30 kb insert, then obviously the amount of nanogram material you need to reach the molarity that we recommend putting on the SMRT Cell would be a little higher. So we recommend maybe like 200 picomolar as the molarity to put on the instrument. So I don't know what off the top of that head is, but I would imagine it's probably only around like 300 nanograms or something like that, or somewhere between 300 and 400 for a 30 kb insert. And that's generally really large. Like most of our libraries, it only takes somewhere between 100, 200 nanograms of material to load at like 200 picomolar, depending on what that insert size is.
Thank you. Another question, can HiFi be applied to bacteriophages? The answer is yes. And there have been, there's been a lot of published work on that as well. So yes, you can use it for bacteriophages. That's all the questions we have in the chat so far. If there are any other questions, please submit them now. If not, we can end a little bit early. I can go ahead and then close it out here. So I'd like to thank the speakers for their time today and for sharing their expertise and experience. Just as a reminder, immediately following closing out of this webinar, you'll receive a short survey. So please do fill that out, as it helps us understand your needs for future webinars. And we did have a question that just came in, so we'll go ahead and answer that then. I just wanted to make sure I got that last part there. So what's the average polymerase read length for this data set, or in general?
So there's two ways of looking at that. We have our HiFi read length, which obviously is that consensus, and I showed you those primary metrics in portion of my talk. And that was like, if you wanted an average, it was like 11.99 kb, just approximately 12 kb. The N50 was closer to like 14 kb. But the actual polymerase, the amount of times it goes around, that's generally like 90 kb or 100 kb. Depending on, on the Revio system, and a little higher, like on a sequencing system. Yeah.
Awesome, thank you. Another question, is the shearing a mandatory part of the high throughput protocol?
Not, no, actually, it's not mandatory. I mean, it depends, right? So, like a lot of times, especially if you're working with metagenomic samples, or maybe say like a gram-positive species, the extraction tends to be a little more, a little rougher, right? You may have to do bead beating or something to kind of really lyse those samples. So in that case, a lot of times the DNA already comes out pretty much sheared around 10 kb. If that's the case, you can just go straight into the protocol with that. If you don't say have a Hamilton technology available, there's other technologies that we have technos for and protocols for. Like there's this company MP Biomedicals that has this fast prep system. That's basically a big tissue lyser. It just shakes like a plate of samples very fast. But what you can do is just load your DNA on the plate, no beads or anything, and it'll just shake it, and that's actually enough to shear the DNA into the size ranges that we need. And there's another company, the Geno Grinder, will do the same thing. So I'd recommend checking out those if you're kind of looking for an alternative high throughput sharing solution that's also low cost. But generally, too, it could also be as part of the extraction, right? If you're doing bead beating like type of extractions, most likely your DNA is already going to come out pretty well sheared.
Great, thank you. Here's a question for John. So how do we go about utilizing the Palmona platform? What's the best approach for that?
Yeah, so the best approach is just once you have raw reads coming back from any kind of sequencing run, so for instance, coming off of a PacBio sequencer or other companies, you can upload those reads directly to our platform. And as I showed in the demo, you can actually then pick different workflows to run on those reads. So let's say you want to do a assembly and annotation bioinformatics workflow. You can do that all via the system, via our web browser, and run through that, and then those results are run in the background in the cloud. It's all cloud-based, and so they can be run in parallel. And as soon as those analyses are completed, you're able to then view and visualize those results. And as I showed too, we have a number of different dynamic, nice visualization tools. So these aren't static visualizations. You're getting back rich dynamic visualizations that allow you to filter through your results and query those results, et cetera. And so we're continually adding new bioinformatics workflows, so depending on your study of interest, but we do have several workflows for assembly, annotation. We have that for bacterial pathogens, viral pathogens, et cetera, and we're continually adding those every day. So you can sign up to join the Palmona platform. Please visit our website where there's more information on subscribing and becoming a user of the platform. And if you have workflows in mind that aren't existing in the platform, we can work with you to implement those. And otherwise, you can also just use several of the workflows we have there.
Great, thank you. Greg, a follow-up question to the shearing question. To clarify, if the samples are long fragments and are not sheared at all, will it work with the high throughput protocol?
Yeah, it depends how long we're talking about. Generally, to get a HiFi read, we want to kind of keep it under 30 kb. If it's longer than 30 kb, your yield's probably not going to be very high because the odds of getting those multiple passes to do that consensus sequence generation is just going to be lower. So we generally recommend keeping it below 30 kb. But by long fragment, you mean under 30 kb, then yes, it's possible. But yeah, generally, we like to keep it on that target.
Thanks. Another question, can you comment on GC bias, if any, for the HiFi prep kits and how it compares to other sequencing technology?
Yeah, I'll briefly mention this, and then I'll let maybe Jonathan talk about what he saw in the data. So yeah, no, there shouldn't be any GC biases. We're not doing any amplification here. So there shouldn't be any GC bias. And Jonathan, I don't know, what did you see in the data? I don't think we saw any of that, right?
No, I didn't notice any GC bias, and again, the results there were consistent with the short read technologies as far as GC percentages, so didn't notice any bias there. Yeah.
Awesome, thank you. All right, it looks like the queue is clear, so I think we'll go ahead and end with only 2 minutes left here. So thanks everyone for joining us today. I hope you'll join us again in a future webinar. And with that, take care and have a great day.
Yeah, bye everybody. Thank you.
All right, thank you.